Good afternoon, and welcome to the USDA Human Nutrition Research Center on Aging Monday seminar series. Before introducing today's speaker, let me remind you to ask your questions for our speaker using the Q&A icon located on the webinar toolbar. Please do not use the chat box. You can ask questions at any time during the presentation as they arise. There is no need to wait until the end of the presentation. And each month this year, our seminar series has featured a speaker from the different USDA ARS Human Nutrition Research Centers. And today's speaker is Dr. Brian Bennett from the Western HNRCA, where he is the research leader for the Obesity and Metabolism Unit and an adjunct associate professor in the Department of Nutrition at the University of California, Davis. Dr. Bennett's research explores the relationship among many types of data, such as genetic variants, gene expression levels, and metabolite levels, and how these interact to increase susceptibility to cardiovascular disease. Today, Dr. Bennett will present his research on the regulation of the cardiometabolic disease-associated metabolite, trimethylamine N-oxide, better known as TMAO, in his presentation titled Aiming for Precision Nutrition, the Cardiometabolic Risk Factor, TMAO. And with that, I will turn the microphone over to Dr. Bennett.
Thanks, Paul. Really appreciate the introduction and the invitation from the folks in Boston. I've been attending these, and they've been a great seminar series. So I think I'm the last of the quarter or the semester, so I hope to not disappoint. And with that, we'll get started. I thought that it would be a good time just to take a second to introduce the center and the group. We're really far away from the Tufts Center, but close intellectually. And so this is an older picture of the center. These are really old cars, and the trees are much larger after it was built in about 2006. And the Nutrition Center here has two units. The unit that I help lead is the Obesity and Metabolism Unit. We have some prominent scientists in the group, including Lindsay Allen, who works on micronutrients and breast milk composition now, Liping Wang, who works on zinc biology, John Newman, who works on lipid mediators and also metabolomics. I know he collaborates with some folks at Tufts currently. Kevin Legiro leads our CRISP project and works on stress biology. Nancy Kime works on diet patterns and energy balance, and then myself down here with an open position focused on alternative proteins. And our group focuses on randomized controlled trials, examining the effects of following a diet based on the dietary guidelines for Americans. That's kind of our main mandate. We work together as a group and focus on some CRISP projects related to that. But one of the things that we know is that following the dietary guidelines, it's a population-based guideline, and the population really people have a difficult time following those guidelines if we look at adherence to the diet by HEI score across the population. And so one thing that has come up is what about precision approaches, or how can we improve adherence or efficacy of our dietary recommendations? I think many of us are aware of a paper by Griffin Rogers and Francis Collins kind of defining this idea of precision nutrition. And in this short paper, they discussed a strategic plan for the NIH research, really to answer a few key questions around what do we eat and how does it affect us? What and when should we eat? How does what we eat promote health across our lifespan? And how can we improve the use of food as medicine? So really 4 very broad but very important topics for anyone interested in nutrition. And one thing I wanted to point out is that synergizes a bit well with some of the goals of our national program in ARS. These are the program components. Looking at agricultural practices to help beneficial health outcomes. And then 2 of the ones that we're really focused on probably in both of our centers, the scientific basis for dietary guidance, which is really what do we eat and how does it affect us? and then what and when should we eat, and then life stage nutrition and metabolism, how does what we eat promote health across our lifespan, with the Tough Center being focused primarily on aspects of aging. So there really is a synergy between what our topic focuses are within ARS and this larger call to action related to precision nutrition. And when I was asked for a title, I had to work in somehow something about precision nutrition, because I've seen it be consumed, as many of us, with working on a project called Nutrition for Precision Health. This is just a nod to the fact that out here in Davis, I'm leading one of the clinical centers and some of the work here in Davis, and really working with great colleagues across the country on this project, including Cy in Boston. And she and I have gotten to know each other much better over the last 2 years, as we've spent a lot of time together working on Nutrition for Precision Health. And so the objectives of the study are to examine individual differences observed in response to different diets by studying interaction between diet, genes, proteins, microbiome, metabolism, other contextual factors. We're going to use AI to develop algorithms and then hopefully validate some of these algorithms for clinical application. One thing that I wanted to point out was that a major focus of this project, and this is just information off the website describing the project itself, is that there are 2 data generating centers. One is the UCSD Microbiome and Metagenomics Center, and the other one is a Metabolomics Center at the University of North Carolina at Chapel Hill. So really, what are the effects of the microbiome, and what are small molecule metabolites that may help us inform the effects of diet and understand inter-individual variation to diet response. And I think this focus from Nutrition for Precision Health was kind of an aha moment for me as I got ready for the talk, just reminding me that, obviously we've all heard so much about the microbiota over the last 10 or 15 years, and the effects of the microbiome on health and disease. And as nutrition scientists, we often really did focus quite a bit more on digestion and some of the other aspects related to nutrition and micronutrient susceptibility. And so right here, I'm just showing a cartoon of just showing that there's dietary components such as carbohydrate, protein, and lipid that are obviously going to be ingested and digested in the gut lumen. There's also drugs, and not only will those things we ingest wind up being metabolized by the microbiome, but they also affect the composition of the microbiome. And during this metabolism, gut microbes that are shown here will wind up interacting with short chain fat, metabolizing the things we eat to create things like short chain fatty acids or tryptophan derivatives, and then interacting with systemic factors. And so on the right-hand side is a cartoon that a graduate student in the lab led a review paper looking at some microbiota phenomenon that we're interested in cardiovascular disease. And so just giving a nod that there are interactions between the gut microbes themselves and aspects of inflammation related to all types of inflammation. Short chain fatty acids also have an anti-inflammatory effect. And then on the far right side, there's something called TMA. And this pathway here is one of the things that we've been focusing on over time. So just to pause again, there's all this information related to the microbiome, metabolomics, what's happening as we're eating our food and it's getting metabolized and digested, but then also it's the effects of those metabolites on host systems and host disease susceptibility. And so the interest in TMAO, it's a link between diet, the gut microbiome, and vascular disease is one of the ways I think about it. A bit over 10 years ago, there was some really nice work from Stan Hazen that identified TMAO as a metabolite associated with cardiovascular disease. This is just a figure from that paper looking at 845 folks without cardiovascular disease that were controls, those with cardiovascular disease, and levels of this metabolite called trimethylamine and oxide. Work within that paper also showed that the severity of atherosclerosis measured by the number of arteries diseased in the heart seemed to increase as well, thereby indicating that there was this kind of relationship between concentration of TMAO and severity of atherosclerosis. Through a series of experiments in that paper and follow-up papers, there was this proposed pathway around the metabolism of TMAO to try to understand this relationship between right here, TMAO and atherosclerosis on the right-hand side. So looking at the left-hand side, there are foods that we eat that contain phosphatidylcholine or carnitine. Those are ingested, digested, metabolized by the gut flora in the gut. And then something called trimethylamine is absorbed and then detoxified by genes called flavin monooxygenases in the liver, and that winds up leading to the production of TMAO, which is associated with atherosclerosis. So it's a complicated pathway involving both diet, microbiome, host, genetics likely, and atherosclerosis. So after that initial paper, there were a couple questions, and I list some of them here. We were really focused on something called FMO3. It's a gene associated with trimethylamine, which is rotting fish disease. So it winds up folks have breath, urine, and sweat winds up smelling like rotting fish. So we were focused on that, and it was a known rare mutation that winds up affecting having that phenotype. And one of the things that they treat those patients with is reducing their choline intake. So we were focused on how is FMO3 regulated, and does modulation of FMO3 expression alter circulating TMAO concentrations, and kind of what are genetic, dietary factors regulating FMO3, and our TMAO levels associated with atherosclerosis in mice. So kind of a few questions we're going to walk through right now. So this is a busy slide looking at some of the molecular biology we did. So we had in cell culture, we wound up creating overexpression constructs, and then looking to see which of the FMOs, there's a family of genes called FMOs. They're all located on chromosome 1 except for FMO5. And so we wound up creating these constructs and then overexpressing them in culture, and then adding deuterated TMA, so we can measure TMA levels, and then also TMAO levels in the media to understand which of these flavin monooxygenases are actually converting TMA to TMAO. And so by and large, this figure is showing that in the center of the left-hand side, we wind up having FMO3 winds up reducing TMA levels, has much higher levels of TMAO. And so we were really confident that FMO3 is the primary FMO affecting generating TMAO levels. And so on the right-hand side is just when we express all of this data as a relative specific activity, it looks like there is some activity from FMO1 and FMO2, but by and large, FMO3 is the main player in this conversion. We also, this is, older data, but it's before things like GTEC. So we went ahead and did some tissue panels looking at FMO3 gene expression by qPCR. And so by and large, most of the expression is in the liver, in the lung, or some lower level expression in the aorta. One thing that I wanted to point out is that the expression, at least in the liver, is very sexually dimorphic, where females have much higher levels of expression than male mice. And so we did some follow-up experiments. I don't have time to share that, where we gonectinized mice and then added either dihydrotestosterone or estradiol, and wound up understanding that hepatic FMO3 and plasma TMAO are inhibited by testosterone. So we're able to kind of look at what are some of the causes of the sexual dimorphism. So once we were focused on FMO3, we really wanted to understand, could we modulate FMO3 gene expression and affect TMAO levels? And so again, looking at some molecular biology and using adenovirus to overexpress FMO3 in males. So males have really low hepatic levels of FMO3. So when we overexpress, you can see a tremendous overexpression of FMO3 that corresponds in the center underneath the males to the protein levels. So there's much higher levels of the FMO3 protein, and then an increase in TMAO shown at the bottom. In females, we did the converse experiment where we wound up using antisense oligonucleotides, knocking down FMO3, reducing the level of the FMO3 protein shown in the center of the female bar, and then wound up looking, there was a reduction in TMAO levels in females. So definitely modulating FMO3 affects TMAO levels. One of the things that I worked on quite a bit as a postdoc was complex trait genetics in mice, and we're using different inbred strains of mice. And so here it's just looking at FMO3 mRNA levels in several inbred strains of mice, BALB-C, BTBR, C3H, C57, Black6, and FEB, on either a high-fat or a high-fat diet with something called cholic acid. One of the things we were interested in was atherosclerosis, and that's an early lesion model. It's it's a diet perturbation that will lead to altered lipid metabolism and some atherosclerosis in inbred mice. And so when we feed mice a high-fat cholic acid diet, there actually is an increase in TMAO levels. So there's an increase in atherosclerosis, and there's also an increase in TMAO levels. So one of the things that's interesting about that is that cholic acid is a prototypical ligand for something called FXR, a nuclear transcription factor that's involved in many processes and widely researched. We were lucky that we were able to work with a colleague named Tom Ballum at UCLA to do some detailed experiments where we use inhibitor, agonist for FXR in both wild type and knockout mice. And so I've separated, males and females are separated here. On the left-hand side is FMO3 gene expression. And we can see that when we use the agonist in wild type mice, we have a dramatic increase in FMO3 expression. And then on the right-hand side, when we use the agonist and then measure plasma TMAO levels, we have a less robust, but a statistically significant increase in plasma TMAO levels in both females and males. And one of the things I wanted to point out is that the knockout mouse is a knockout for FXR, so it's a proper negative control where the agonist is not having an effect. There's no off-target effects here happening. So one of the things that I wanted to do right now is, so I've given you a little bit of information about FMOs and the fact that FMO3 is one of the critical mediators and players of TMAO biology. But there's this other microbiome component. And so this is going to be kind of a hard shift, I guess, as we walk through the different aspects of the pathway. And I just wanted to acknowledge some of this work, there's too much to fit into a talk across all the wonderful studies that many people have done. But really, one of the things that I like about this pathway and where some of the research is headed and some of the things that have been found is that we've moved a little bit away from relying only on sequence-based methods to look at interactions. There are many ways to do these studies, and I'm just going to outline and show three kind of examples of how people have done this work, where they've either used conventionalized mice and either a diet perturbation, a specific inhibitor, or engineered bacteria to affect phenotypes in mice such as TMAO or atherosclerosis. Other folks have used either germ-free mice and done some fecal microbiome transplants. or done similar sorts of studies in germ-free mice. And there are many ways, I kind of lump together germ-free mice with being gnotobiotic, or those that have been exposed with to antibiotics to knock down the microbiota. So these are just three broad ways that people do this work. And I just wanted to show one piece of like a little vignette of part of the story around TMAO, which is when we were working with many different inbred strains of mice, we identified two NZW, which was highly resistant to atherosclerosis. So on the left-hand side of this graph, you can see atherosclerotic lesion size, and plasma TMAO is on the x-axis. And so this is just a correlation between those two phenotypes. And what we wind up having is that NZW have fairly low plasma TMAO values, really low atherosclerosis, and C57 Black 6 is susceptible to atherosclerosis, and actually has pretty high TMAO values. So one of the initial studies that that was done, and so this data on the left I helped generate, on the right was some work that Stan then took, and his lab wound up performing some experiments looking at C57 Black 6 mice and NZW, and looking at plasma TMAO values after a fecal microbiome transplant. So they took Black 6 mice, transplanted in either Black 6, C57 Black 6 microbiota or NCW, and then looked for TMAO values over time. And so NZW surely did have lower TMAO values. So that, again, having horizontal transfer of this trait between strains, and it all being mediated by the microbiome. They did a series of experiments that were published several years ago, but just kind of showing some of the work that they did do, where they go, they have a fecal microbiome transplant, so they wind up having antibiotics for 3 weeks to knock down and ablate the microbiome, and then they gavage daily for several weeks, and then wait 11 weeks to quantify atherosclerosis. And so the donors were C57 Black 6 mice, or NZW, into something called an ApoE knockout mouse, and an ApoE knockout mouse is an atherosclerosis model. And so what they see here is that there's when you wind up recapitulating or reconstituting the microbiome with NZW, you wind up having very low TMAO values, and when you reconstitute with C57 Black 6, you have much higher values. So ablating the microbiome reduces TMAO values, it decreases atherosclerosis. That was some of the other work that Stan had shown prior to this. So it does look like at least severity of atherosclerosis and TMAO values can be affected by the microbiome, and that it's transferable between donors and recipients. So Stan has spent a lot of time focusing on the microbiome, looking for specific inhibitors and pathways within the microbiome affecting the conversion of dietary choline to trimethylamine, and has several years ago published some specific inhibitors on this. So I'm not going to walk through this. It's not really necessarily something that my lab's focused on, but we are aware, and it's obviously great work to follow along in the literature. And I'm going to switch now to trimethylamine N-oxide's diet component. And so that's kind of this circle here. So we're kind of working left in, towards the left in this diagram. So there are two nutrients that have been associated with TMAO. The first is choline. It's an essential nutrient. Work with Steve Zizella has shown many different beneficial effects of choline on growth and development, and brain development in particular. It's got important roles in lipid biology, and also as a methyl donor. it's got a relatively high adequate intake number, and often it's one of the nutrients that Americans can be a little bit lower in or more susceptible to deficiency. And then on the right-hand side, there's carnitine. It's a non-essential nutrient. It's got a role in fatty acid oxidation. And both of these are primarily found in animal-based products. And here's a table that we pulled, just kind of given an idea about how much choline and carnitine is in kind of what we would consider a normal serving. And so things that Americans, consume on a regular basis, beef, eggs, milk, salmon, maybe a little bit less than we should. So knowing the nutrients that are going to lead to perhaps TMAO values or things that are associated with TMAO, there's been a number of studies investigating different dietary interventions affecting TMAO values. And the one on the left here is some work done by Zheng Wang and Ron Krauss several years ago, where they wound up having a fairly large clinical trial where they were feeding individuals different types of meat, either non-meat, red meat, or white meat, but all the diets were isocaloric and it contained 25% of their calories from protein. And so in this work, what you're able to see here is that when folks were consuming the non-meat diet or the white meat diet, they had relatively low levels of TMAO. When they were consuming the red meat diet, they wound up having quite a bit higher levels of TMAO. And so that's the center AB part. And so definitely, really good clinical trial showing that at least in their hands, consumption of red meat led to increase in TMAO values. So that was a longer term 4-week study. And then there were also a flurry of activity around acute feeding trials and responses to TMAO. This is a really interesting paper by Marie Caudill's group several years ago, looking at fruit as a control, egg, beef, or fish, and TMAO values. And this one was really interesting because the triangles that are shown here and the TMAO values that are shown that you're seeing are for fish. So they fed healthy volunteers a fish meal and followed them over about 6 hours, and you see a dramatic increase in TMAO values. So fish use TMAO as an osmolyte, especially deep water fish. And so when people are consuming fish, they actually are getting a pretty good dose of TMAO. And I always acknowledge this one because this is the one data that really kind of stumps part of the nutrition recommendation part, where for heart health in AHA, there's often a suggestion to eat more fish and things with omega-3 fatty acids, such as salmon. And then there's this other data that shows that acutely fish winds up raising TMAO values and way, way higher than beef or egg. So in this study, actually, egg and beef did have an effect, but you can't see it just because it's dwarfed by the effect of fish. Oh, and just before I move on, I just wanted to point one thing out, and I think we're all really aware of this in our nutrition trials, where this idea of responders, non-responders, and in some cases hyper responders, folks who highly respond to a diet treatment. And we see that here in this data, and I think many people have seen that across many studies with TMAO. So similar to others, I wound up arriving in Davis and started working on some secondary analysis of TMAO in different clinical trials that we wound up having available to us. The one on the left is a Mediterranean style eating pattern with different levels of red meat. So the low red meat diet is 200 grams of unprocessed lean red meat. The high red meat diet is 500 grams of unprocessed red meat. And so what we wound up seeing was when subjects were finished the low red meat diet, they actually had lower TMAO values than prior to starting that or when they were on the high red meat diet. There really didn't seem to be a difference in the red meat, the TMAO values when they were fed the 500 grams versus any other time. So it looks like when we reduce red meat consumption, you can actually lower TMAO values. So that was interesting work led by Sridevi Krishnan. I have a picture of her up here. She was a junior faculty member at the WHNRC and UC Davis, and has started her tenure track position at University of Arizona recently. And so beyond that, we also had finished, right as I arrived, something called IMAPS, which was the test of the USDA's dietary guidelines and anti-Kime led. And so in that study, it was a traditional American diet or typical American diet versus a dietary guidelines diet. It was an 8-week feeding trial, really well done. And what we're really seeing here is no effect. We saw no detectable effect between week 0 baseline and then the completion of the trial, which was a little disappointing. We really thought there would be, but it was a relatively small study, and I think the literature has borne out that these smaller studies, the hyper responder and non-responder phenotypes, not knowing what to select the population for, and this being a secondary analysis, you do get a little bit of noise, especially, and we see that in week 0 of the DGA-fed individuals. So plasma TMAO is associated with several foods and diet patterns, initial associations between eggs and meat and short-term meal-based exposures. Many associations-based studies utilizing food frequency questionnaires, ASAs, 24s are the recall data from cohorts and then looking at TMAO values. The data is not completely consistent in those studies. I just wanted to spend a little bit of time focused on some of our most recent CRISP projects. This is something called the nutritional phenotyping study, and it was a 2-visit study with about 400 participants where we had subjects come in on a weekday, get trained on how to do ASA 24 and some other things related to the study. And then on visit 2, they would come in and do a longer test day where they were extensively phenotyped, given a meal challenge. And so I had a graduate student in the lab, Kristen James. She led these analyses, and this is some of the work we were focused on are, what foods are associated with TMAO, what microbes, what genes, are there any interactions, was TMAO associated with vascular function in healthy humans? So this cohort was non-medicated, relatively healthy individuals. We recruited into bins over time, and the bins were age, sex, and BMI. So we were trying to balance recruitment over time to not have skewed data. And so what we see here is that there's TMAO varies by age, and that's shown in the graph here, that as subjects wind up becoming older, from 18 to 33 was our lowest age folks, and the 50 to 65 was the maximum age. We wound up seeing an increase in age, and there was also, I'll just note, an increase in TMAO with or an association with cystatin C, which is a marker of renal function. So intake of choline and carnitine containing foods, this is, from our ASA 24 data, and, after kind of curating and cleaning and doing all of that work, this is the intake of foods. We didn't necessarily see a lot of association between our diet intake and TMAO levels. And one of the things that we realized is that, at least in our cohort that was primarily based here in Davis or close by in California, the actual intake of red meat and other foods is a little bit lower than average compared to the rest of the US population. And the other thing that we were doing was recruiting healthy individuals, and TMAO values were relatively low in the cohort overall, because most of the individuals were screened for things like lipids and drugs, or medications for blood pressure and other metabolic disease. So it was generally a very healthy cohort. So there wasn't much of an association between diet and TMAO. We also obviously looked at the microbiome, as it's so important. What Kristen wound up doing was breaking up the cohort into tertiles and then looking at progressively increasing levels of TMAO to see if there were associations with the microbiome. Unfortunately, they were subtle, and there was an association between diversity and TMAO values. But overall, the cohort is relatively healthy, and the associations were modest, at least in our hands. And the microbiota analysis was run by one of the ARS scientists here that's in the immunity disease prevention group, Mary Cable. She's a collaborator on a lot of our microbiome stuff. She's a microbiologist. And so Kristen was banging her head, doing a lot of analyses related to some of our initial thoughts around endothelial function. We were using some clinical-based measures of vascular function, plasma chemistry levels. But the one thing that really popped out was an association between TNF alpha and TMAO. So these are tertiles of TMAO. So at the top is the range of TMAO, up, really low, medium, and then actually a fairly wide range in tertile 3. And so what we see is a pretty strong association between TNF and TMAO. It's one of the few things that we actually saw related in this cohort. But being interested in genetics, one of the things that I was interested in was a couple of the SNPs in the FMO3 gene that have been proposed to affect the conversion of TMA to TMAO and related to the fish malodor syndrome. And so what we have here is data split by males and females. So on the left-hand side is males, and this one SNP, RS2266780, winds up having really no effect in males. In females, we saw this interesting relationship between the AA alleles, or females carrying the AA allele, and age. So TMAO levels seem to increase in females carrying this particular SNP. So it's definitely exploratory analysis. It's a small cohort, but does lend itself to wonder what are the causes of this, or what are the implications of having this SNP in females. So one of the next things we did was glucose and fat tolerance test in FL100. I initially thought that this wouldn't work, but I was encouraged by Kristen that there was this paper from in Nutrients a couple of years ago showing that oral glucose tolerance test and oral fat tolerance test could modulate TMAO, and that data is kind of shown right here on the right. So what Kristen wound up doing was taking the nutritional phenotyping study and then doing some fasting blood draws, or looking at the TMAO values in a subset of individuals, about 100 over time from the mixed meal tolerance test. So these are the values at 30 minutes, at 0, 30, 3 hours, and 6 hours. So that was kind of the time points we looked at in the meal challenge. And then what she wound up doing was classifying them based on what time their peak TMAO values were. And so it looked like there was a subset that were at peak at fasting at 30, 3 hours or 6 hours. And then related that to a number of the phenotypes that we have on the left-hand side are the fasting values on the y-axis. On the x-axis are the area under the curve values. And what we can see is that for individuals whose peak TMAO is at 30 minutes or 6 hours, they have a pretty strong association with fasting TNF values. We also had TNF across the meal challenge, and that relationship between area under the curve for TMAO and area under the curve for TNF remain relatively robust. So it looks like there's something about this mixed meal tolerance test and TMAO values and a low level circulating levels of inflammatory mediators like tumor necrosis factor. So one of the things that we're really focused on is overall genetics. And I'm going to just focus for a minute on our work on genetics. So some of this work I did with a close colleague, Houman Ali, who's at USC. And our first thought was, well, we better like interrogate the FMO3 locus. And so FMO3, we could identify SNPs near the FMO3 gene that affected the expression of FMO3, hence an eQTL or expression QTL. But when we looked at plasma TMAO values in about 2,000 patients from Stan's GeneBank cohort, we didn't see any genetic signal. And then moreover, when Houman went into the cardiogram consortium and looked for signal at the FMO3 locus, there was really no association between FMO3 and cardiovascular disease in that really large cohort. So the story is a little bit more complicated than just focusing on FMO3. And so we did a genome-wide association in something called the Hybrid Mouse Diversity Panel, that the results are shown here. And there was a hit near a zinc transporter called SLC30A7. But in humans, we saw a very different signal, and this is actually an expansion of the initial work. And we're not really seeing anything that's getting across a significant threshold. So we've been focusing a bit in my lab on the mouse work. And one of the things that we did is we shifted to something called the diversity outbred mouse population. It's a combination of 8 inbred strains of mice. They're listed here: AJ, Black 6, 129, NOD, New Zealand obese or NZO, Castaneous, PWK, and WSB, the latter 3 being wild-derived mice. And so Jackson Lab is inbreeding these and creating something called the collaborative cross, or taking partially inbred collaborative cross mice, and they made something called the diversity outbred. So a family of about 175 mice that are mosaics and combinations of these 8 strains of mice. So they're randomly mated, and you wind up shuffling the genome over and over again, making it really good for genetic mapping and other things like that in complex trait genetics. So these 8 strains are shown here just to give you an idea about what they look like, not in a cartoon form. And we wound up phenotyping these mice on different diets. And one of the things that's different, shown in panel F here, is that TMAO is different between the inbred strains, again, suggesting that TMAO will be under genetic regulation in this population. And so these studies often wind up having a 2-week lead-in diet where we have a standard AIN diet. We phenotype the mice, and then we put a diet perturbation or some other perturbation on the mice, and then look for response to the perturbation. And in this case, we focused on 2 diets: this high-fat cholic acid diet and a high-protein diet. And we had about 300 mice, and we wound up doing some transcriptomic analysis, looking at gene expression and small RNA-seq expression. This is work I did when I was at the University of North Carolina, and I'm just acknowledging Bean Sethampathy is a microRNA expert, and he and I were kind of trying to understand what could be regulating FMO3 expression, or how could this work? And so we have the 2 diets. A high-fat cholic acid diet does increase TMAO levels, that's shown here on the left. And then we looked at small RNA-seq sequencing and found that miR-146A was this highly correlated microRNA with TMAO values. And so we were pretty excited about that. And we wound up looking at TMAO values in a couple of replication cohorts. One was African green monkeys. We measured TMAO in that study. That was a study that Ryan Temmel ran, and then correlated that with miR-146 expression. And so sure enough, it does look like that relationship replicated in non-human primates. We did some other studies with something called Lurco mice. I don't have time to describe that to you now, but in all 3 cohorts, the DO mice, the African green monkey, and the Lurco mice, miR-146 was associated with TMAO. But where this got complicated was that miR-146A isn't associated with FMO3. So there's not that it's not associated, there's not a miR-146A binding site in FMO3's 3' UTR. So any interaction or any regulation that FMO3 that has from miR-146A is going to be indirect or through some sort of complex relationship. But I told you that the DO is very good for working on genetics. This is one of our initial gene mapping results from that initial study. And I point out that there's a locus on chromosome 12 containing 80 genes, and that's shown here. And these other things are just kind of the log of the odds ratio is the measurement here. Anything above the red line is significant via permutation testing. So what we can do with trimethylamine N oxide is go ahead and zoom in on chromosome 12, which is shown here on the left-hand side. And then because we know all the SNPs in those 8 strains that we've used to make the DO mice, we can recapitulate haplotypes and say, which of the founder strains or which of the alleles from the founder strain seem to be associated with the phenotype. In this case, it looks like the green and the red on the left-hand side are associated with lower TMAO values. That's the Castaneous and PWK haplotypes, so those part of the wild-derived genomes. On the right-hand side, we also could look at gene expression for genes at the locus and then try to understand if there are genes that are correlated with TMAO. But we could also ask, are they also regulated by the same SNP that we're seeing associated with TMAO? And that's a positional candidate of these 80 genes was a gene called NUM. So this is just showing how we kind of relate both the clinical phenotype on the left-hand side and then the more complex gene expression data on the right-hand side. So while we were working on this, we wound up having multiple studies that we were working on TMAO or phenotypes related to TMAO. And so Kristen, who is a really motivated grad student, not only did the human work, but then wound up working on some of these analysis of genetics. And so what she wound up doing here was replicating the TMAO locus in 5 independent cohorts, 2 from UNC, and then 3 that were worked on here at UC Davis. And so what you can see from the blue line that's kind of popping up over chromosome 12 multiple times is that this QTL is replicated. And that's really was really encouraging that we're seeing kind of a robust genetic signal across multiple studies in multiple locations. And on the upper left-hand side is kind of the result of what happens when you perform a meta-analysis in all of these data, is that there's a really strong QTL on chromosome 12 with a LOD score of about 60. So we're really confident that there's something here on chromosome 12. And just giving a nod to the fact that we're being thorough around looking at sex as a biological factor. This is the QTL replicated in both males and females, even though males have relatively low levels of TMAO. And so what you can see here is that this data on chromosome 12, the green are males, the pink is females, and the all is blue here. So kind of breaking down that QTL by sex. And then on the right-hand side is the genetic effect at the locus on chromosome 12 for the lead SNP. So you wind up seeing a kind of an additive effect of genotype at that lead SNP. Here's a list of some of the genes in the locus, and then this is actually their allele effects. So this is one of the ways we've been trying to understand what are positional candidates that whose genes are expressed, at least in the liver. And so we're not really ready for prime time. We're thinking about what are the follow-up studies that we're going to go after these specific genes, and how will we understand which gene or genes are affecting TMAO at the locus. And just giving a nod to some extension of the work that we're doing in diversity outbred mice is that we're actually trying to modulate choline levels, either making them deficient or or hypersupplementing with choline and looking at TMAO values and seeing how that affects the genetic regulation, looking for essentially a gene by diet interaction. So here are some conclusions. One potential mechanism for microbiota to affect host physiology and pathology is via metabolites. That's kind of this idea that's getting really well tested in nutrition for precision health. And the example that we're focused on in our laboratory is TMAO and atherosclerosis, and kind of detailed studies there. Diet effects on TMAO have been varied. Acute increases in TMAO following intake of fish, beef, and eggs. And the diet pattern work varies. Increases in meat intake does seem to increase TMAO in general, but there are a lot of responder and non-responder phenotypes in most of the diet trials, and especially some of the secondary analysis has been a little bit harder to tease out the consistency and reproducibility of diet effects on TMAO. So TMAO changes in a meal test, and it's a variable response of that. And one of the things that was really interesting there, again, just to reiterate the point, is that depending on when a person was able to metabolize TMAO, there was an association with circulating levels of TNF. And so there seems to be a connection between TMAO and inflammation or TMAO generation following a meal and levels of TNF. Genetic signals for TMAO have been difficult to find. The mouse studies have been very helpful to identify genetic regulation and potentially novel gene targets. There are potential candidates on chromosome 12, and miR-146 seems to be associated with TMAO levels. Understanding, which genes miR-146 may be interacting with is another way that we're kind of sorting out candidates at chromosome 12. And so the last thing that I'll have to say is, that every once in a while there's a question about, should we change our diet based on TMAO? And kind of one of the things that I think is that it's too soon for a precision recommendation. I don't think we know necessarily enough about the underlying genetics, the variable response to meals, or the microbiome effects to know, should we be changing our diet recommendations for specific individuals. I don't know that that's the case. I think obviously for folks that have the fish malodor syndrome, they're already, often seeing a physician and being recommended to reduce their intake of choline-containing foods in order to mitigate the effects of carrying that mutation. But beyond that, for the general population, I think we're a little bit too soon. So with that, I just want to take a minute to acknowledge, a lot of the folks that I've worked with on these studies that I've shown you and other studies related to TMAO and precision nutrition. Have a great group of folks here at the WHNRC. In my laboratory, I've worked with, had a lab at UNC, and some of the work that I share today was either started there or was completed there by Anna O'Connor in particular, or Tanji Smallwood and Alicia Coffey. And then at the Human Nutrition Center, working with Nancy, Mary, Lindsay, and John, I've been able to get a lot of support locally around finishing some of these studies. And then we have a number of collaborators here at UC Davis, UNC, or across the country that we've worked with in order to kind of understand a little bit more about TMAO. So with that, I'm going to pause. I think I may have rushed through that last part around genetics. I got a little bit nervous, but with that, I think I can take questions.
Okay, thank you for a very interesting talk. TMAO discussions are always quite fascinating. So we do have a few questions. First question comes from Richard Alpert, who asks, given the acute rise in TMAO after fish consumption, why is fish popularly represented as a heart-healthy food?
Yeah, and this is always the hardest question. I think that there, things like fish oil, and other fish have been recommended to reduce cardiovascular risk factors and, incidence of disease, prior to TMAO being identified as a risk factor. So that one's a little bit harder to understand. I think part of it is going to be that it's an acute rise in plasma levels following ingestion of the meal, but I don't know that people have tested this. If you continually eat fish, does your fasting TMAO levels change over time? And so the association that Stan has shown and others have shown is between fasting TMAO levels and cardiovascular risk. So I think that's one aspect. And then the other aspect that's equally important, so it's kind of, I guess, clearance by renal function to get, that acute load of TMAO out. So, I think that's one of the harder ones to reconcile. So, Richard, I hope I answered your question. If not, just put another one in there.
Okay, next question comes from Saidas. Great talk, she says. Is there work in females during the perimenopausal and early postmenopausal periods for changes in gut structure and flora and FMO3 and TMAO, or even in mice where menopause is chemically induced or age equivalent to humans?
Yeah, it's a great question, and so there have been a limited number of reports about I guess menstrual cycle and either the fish malodor syndrome in specific populations. So there could be something going on with sex hormone biology during menstruation. So that's not something I've focused on, but we're kind of aware of that work. And then the menopause stuff gets pretty interesting because we see this age by genotype effect, and there's kind of smoke in the air. I'll just say, Cy, where it winds up looking like there's an increase in cardiovascular risk in females after menopause. There looks like there's an increase in TMAO based on genotype with age. So there is something maybe worthwhile following up on there. I'm not sure that anybody's looked at it that way, but it does kind of lend itself to wonder what are there effects of aging in women that we need to be a little bit more focused on and related to either sex hormones and interactions with FMO3, or as I suggested, kind of the changes in the microbiome that may be impacting the conversion of foods we eat to TMA.
Okay. Michael Lustgarten asks the next question, which is, how does soluble fiber intake impact circulating levels of TMAO?
Yeah, that one's another little bit of a stumper. So there was a postdoc who worked with Ron Krause who did a fiber intervention, and really kind of counterintuitive to the way I would have thought of it. Again, fiber being quote unquote a good food for you, wound up increasing TMAO values in her study. And so I'm not exactly sure how to think about those data and what types of fibers may be affecting expression of genes or I guess populations of flora in order to increase TMAO values. So that one was harder to understand, but it's something that's reported. I don't know if there's been more comprehensive work looking at either fiber type or replicating that result in particular, to understand if there was something different about that cohort, or if it was something different about the fiber that wound up pushing TMAO values up or down. my a priori hypothesis before that would have been that, increasing your fiber in your diet would have actually reduced TMAO, but that's kind of where that landed.
Next question comes from Chris Wiley, who says, great talk. How much of a challenge is it translating TMAO findings between mice and humans, considering differences in diet, atherosclerotic responses, et cetera?
Yeah, Chris, obviously asked an important question for anyone that's doing translational work and using mice in preclinical models. Mice are great models. We can learn a lot from them, but it's not a one-to-one comparison. There's a lot, especially in atherosclerosis, we use often gene knockout mice or other models to induce atherosclerosis, because mice actually tend to be resistant to atherosclerosis. So we get very context dependent in some of the preclinical work. One of the things that obviously is a big advantage is how much we can control in what else is going on. So flavin monooxygenase 3 has a pretty, and a lot of the FMOs are detoxifying enzymes in the liver. One of the things I didn't talk about was that they're not necessarily the best genes probably to target for human intervention. They have really wide substrate specificity and are important in detoxification. So we wouldn't want to necessarily muck with that too much. The reason why I mentioned that is we and others have spent a lot of time looking at FMO3 knockout mice or modulating FMO3 expression to show that this phenotype is possible, but I don't necessarily know that there's a drug target. So I think that's one aspect, like, what's going to be clinically actionable. And then with the ability to control things in mice, obviously we don't have to worry about exposure to environmental pollutants or other things like that. And that is actually one of the ways that FMO3 is induced is environmental exposure. So those are kind of some examples where I think they're just different. And I'm just going to leave it there.
Okay. Next question comes from Jiandong Wang. He has a bit of a lead-in, so bear with me. I'll get to this. It's a short question, but a long lead-in. So TMAO is mainly eliminated greater than 95% through renal excretion. So the link between TMAO and CVD risk is substantially mediated by renal function as a potential confounding factor. You mentioned that certain diets, example fish and eggs, increase plasma TMAO depending on responders or non-responders. And you have also looked at cystatin C as a biomarker of renal function in your study. I was wondering if TMAO was associated with renal dysfunction and TMAO is a biomarker of chronic kidney disease.
Yeah, it's a good question and a really important point. So we're looking at biomarkers in the blood. Renal function is going to be critically important to understand, because if you can't clear something through the kidneys, it's going to actually rise. And so renal function, there's been a few studies, actually quite a few studies looking at renal function and TMAO, preclinical models looking at renal fibrosis and the fact that TMAO seems to be associated with that. So answer in a word is yes. And my understanding through some work that I do with a colleague here at UC Davis that's a renal doc is that TMAO is really high in renal patients, especially those undergoing dialysis. So when there's really poor renal function, you're going to have high TMAO values.
Okay, still a few more questions here. Next one is from Sheldon Rowan. And he asks, in the mouse genetic experiments, are there differences in microbiome composition that influence TMA production independently from conversion of TMA to TMAO?
Yes, and so Sheldon, there's been, and this is one of the things that we get really careful about is that between cages you can wind up having differences in microbiome composition, that could affect the microbiome and hence TMA production. There's a few genes that have been typed by and a few pathways, cut C, cut D, that seem to affect TMA generation by the gut microbes. And I don't think that every bug has been actually identified that contains those. And likely there are other genes in microbiota that help with the conversion of choline or carnitine to TMA. So there's probably a lot more to be learned there.
Next question comes from Chao Lai, who asks, there is no obvious genetic variation associated with TMAO. Have you examined the gene-environment interaction on TMAO in humans?
We haven't in that initial cohort. One of the things that Human's been really beating the street for is, to get a larger cohort. And he's about, I think, up to 40,000 individuals. And so he's kind of working on that angle, hopefully finding, a target, a candidate gene for TMAO. I think that, looking for the interaction is a really great idea. I don't know, and I'm going to talk to him about that suggestion, because depending on how those cohorts have kind of been assembled, we may or may not have enough information for understanding the environmental interaction. But that's a good suggestion. Thank you.
And the last question comes from May Vives, and who asks, I would like to ask if you could clarify what does it mean by in quotes TMAO, changes in meal test variable timing of peak response, which was in the conclusion slide. Does it mean that TMAO production varies according to the time of day the food is consumed?
No, so I guess in our hands, May, thank you, and I probably misworded that on the conclusion, is that in our hands we were performing a meal challenge test, and it contained no choline or carnitine, and so I didn't really think it would push TMAO values that much. And so participants came in a standard protocol where they did a fasting blood draw. About 10 minutes later, they ingested this meal challenge. And then we looked at 30 minutes, 3 hours, and 6 hours at TMAO. And so what we noticed was that there were individuals whose peak TMAO values were prior to the meal challenge, 30 minutes, 3 hours, or 6 hours. So the metabolism of this meal challenge and the appearance of TMAO varied across the population. And so that likely reflects differences in the microbiome, but also probably the efficiencies of some of the enzymes, such as FMO3 or other things that are happening.
Okay, well, thank you very much for a wonderful presentation today. Really enlightening. I appreciate it.
Thank you, Paul. I appreciate it.